Thank you very much. So I will introduce a phenotypic species definition for genetic improvement of source code. In this talk, I will briefly introduce the context of our research. Is it too close? Okay. So I will introduce the context of our research, emergent systems, and how genetic improvement for source code is involved in this scenario. And finally, we need to clarify why we need species. Once we agree that we need the notion of species in this research, I will introduce the challenges related to the genetic improvement specifically and what we propose as a phenotypic definition for species. Finally, I will show some examples. So emergent systems are those that have a skeleton, a blueprint that specifies the functionalities and their interactions without any actual content. Instead, we assume that each functionality can be represented by multiple such building blocks. They provide the same functionality with different characteristics related to speed, cost, memory consumption, and so on and so on. And in each step, we can select between those candidate components according to the given circumstances in the environment. So all these are valid and possible configurations for this system, but nothing is predefined. Everything is configured and reconfigured at runtime. It is a highly adaptable system, and it assumes that we have a set of interchangeable and hot swappable components. Now, this is a crucial question. How will we going to have a set of potential candidates for components? The answer is we simply breed them by genetic improvement. So we channel the input of certain components in this system to a genetic improvement system. We provide the candidate codes in this pool of individuals. And then by genetic improvement, we try to create individuals that are best or better tailored to the given circumstances. And finally, we have a set of them. Now, why do we need species here? The answer is there are two crucial things here. One is, how can we decide that we have a sufficient diversity in this pool? That is, the candidates are sufficiently different from each other and not just little tweaks on essentially the same code. The other point is that the search space in this type of genetic improvement is extremely vast and rugged. Therefore, we need some waypoints to navigate our search in the search space. So we have an idea why we need species. How can we go on? First, I will not tell you any definition for species because there exist over 30 definitions from biology to philosophy, anything. In the most general sense, species are a group of individuals. This abstraction is above the level of individual and below the level of population, and they are similar to each other somehow. The motivations are several-fold in literature, usually tied with multimodal optimization, parallelization, reducing competition. In our case, it's simply twofold. We would like to capture meaningful diversity in our population, and we would like to navigate the search space efficiently. Now, how does GI for source code work? Individuals in this model are pieces of code representing a functionality that we would like to improve. And randomly, we apply genetic transformations like mutation, which could be a modification of an existing part of this code, like a constant or an operation or a condition, or Can we somehow cancel this tag here? Oh, okay. Okay, thank you. So we can insert lines of code or delete lines of code. Plus we have a crossover, which is simply transferring lines of code from a different individual. It is a horizontal gene transfer that is in our model. We don't have the notion of gender and anything like that. It's simply transferring genes between individuals. Then after the transformation, the fitness is evaluated for each individual. And finally, we select the best ones for the next generation. Now, why is GI for source code intrinsically different from any other type of genetic programming? So the genotype in this case is the source code itself, and phenotype is anything that is manifested or observed at runtime. It could be speed, it could be the meaning of the program, memory consumption, energy consumption, anything. So let's have an example here. We have two pieces of code. They differ only in one character, which means their genotypes are very close to each other. Can we say anything about their phenotypes? Supposedly, they will behave similarly in terms of speed. But do they exactly the same? No, they are different. They are so different that they have different names. One is the Bernstein hash. The other one is the Kernighan-Ritchie hash. Okay, let's see another example. Again, just one character difference. What will it do? What is the phenotype? Essentially, it's nothing because it's a compilation error. It will not compile. Okay, then let's take another example. Now it will compile finally, but it's still not proper. It may give runtime error due to array boundaries. And finally, something entirely different, very loosely related to any of the previous codes. What does it do? It is exactly the Bernstein hash. It does bit by bit the same as the first example, yet so the meaning of the code is the same, yet their performance may differ. So there are two important lessons we can see here. One is the genotype and the phenotype are very loosely connected. Minimal changes in the genotype can create dramatic differences in the phenotype. The other point is that the phenotype essentially captures two things: what the code does and how it does it. For instance, in this case, the non-functional phenotypes may be similar, but the functional phenotype is different. In this case, the functional phenotype is exactly the same, yet the non-functional phenotypes may be different. So its phenotype is always a dual matrix. So this is what we have so far, and how can we rephrase this challenge? The genotype encodes the algorithm and its implementation, in a sense, how well the algorithm is encoded. And both they are observed as phenotype. Now, the real question is, if we observe the phenotype, can we infer what the algorithm is? In other words, if we have a very rugged and vast search space where genotypic speciation is not really possible, if we group together individuals with similar genotype that are close to each other, that their phenotypes may be very, very different. Instead, what if we try to find groups on the phenotype dimension? If you are able to find a metric that is selective, characteristic, selective meaning that it is able to detect the smallest difference between two individuals. Characteristic means that it will give always the same categorization for the same algorithm. Insensitive to implementation and easily computable. If you are able to find such a metric, then we would be able to separate individuals by the core algorithm. This is the challenge here. And can we do it? Spoiler alert, we can. At least we believe so. It has two important cornerstones. One is capturing the fingerprint of an algorithm. So phenotypes can be non-functional, like speed, energy, efficiency, memory consumption, and so on, and so on, and so on. And functional, what it does, what is the core algorithm of the individual. Now, capturing the fingerprint of the algorithm is application dependent. We demonstrated our method on a hash example. We can extend it to a larger set of optimization problems, but still it's not universal. We have idea how to improve it and how to extend it, yet we are not yet there. So the essence here is we try to capture some measurable phenomenon here that has a pattern or a multi-dimensional nature, and we can assign a distribution to it. Like in case of a hash function, it is a hash layout. That is, in this example, we have 10 buckets, and we simply count how many elements are put into that particular bucket. How can we compare two such distributions? The solution is to compare them to one known and fixed distribution, like the uniform distribution. For example, it could be anything else. The other cornerstone of this solution is how we measure the divergence of two distributions. Again, it is just an example here. Such probability divergence metrics exist, numerous of them. This Kullback-Leibler that we applied is just an example, and I won't explain the details. What is important here is it is easily computable. So from all these, how do we do the speciation at runtime? We observe, but 2 minutes, okay. We observe the phenotype attributes at fitness testing. We choose some functional attributes like the distribution of a hash table. We approximate the probabilities by relative frequencies, and then calculate the probability divergence, and probability divergence defines the species. So finally, I will show you some examples. Essentially, we would like observe, control the evolution, and then finally find more complex strategies to govern the evolution. This is more or less future work. So here we can see how species are spawn, living, declining, and extinct. Here we start off with the green species. The middle of this patch means the best fitness for that particular species. We can see it is large initially, and then it starts declining because we have a better species, the green colored, which starts growing, but it cannot really gain a mass as we have better candidates here, and then again better candidates until finally we reach this pinkish species that will gain a mass and it will dominate the population. Here we can see the same temporal behavior of the species. Then here we compare the fitness improvements and the existing number of species, and we can see that really big jumps, really big improvements in fitness, which is runtime simply, so the lower the better. They happen when a new species appear here. And we can also quantify this phenomenon by the Shannon and Simpson indices. The Simpson index shows here that despite we have a large number of different species, a few of them is dominant, really. And this is a summary of the previous slide. If we record the best fitness during the evolution, and we see if the best fitness was caused by a new species or existing species, then we can see that existing species cause much more fitness improvement than new species. Yet, the gain of speed or the gain of performance is much significantly better with new species. That is, real changes are introduced by new algorithms and not just tweaks on the existing algorithms. This is a categorization of the species. What is the best fitness measured? We can clearly see a tendency here. And as we approach the zero, which is the uniform distribution, we can see that it is not the goal to reach the uniform distribution because it is getting worse and worse. It is also important to notice that species and performance are orthogonal metrics of the same thing. So species are not tautology for performance. They are essentially two coordinates. If we fix and given algorithm, it can have a very good realization and it can have a poor realization. There is no one-to-one relationship. It is even more, yes? Yep, I will finish it. So it's even more visible here with the median of the fitnesses or with the apex species. These are the very best species in one experiment, but they can be very poor species. The exact same species can be very poor in a different experiment. How is it possible? It is a good algorithm with a poor implementation. The duality of the phenotype appears here. Okay. Essentially, that's it. So thank you very much.
Great. Thank you.